Hello, and welcome to another edition of Ask Excite. I'm Tim Bushnell with Expert Cytometry, and today's question comes from Joanna. And Joanna asks, what are some important considerations for preparing cells for cell sorting experiments? Great question, Joanna. We're going to have to think about where our cells are coming from. Are they a liquid tumor or a liquid sample like peripheral blood, marine water, something of that nature? Are they coming from an adherent plate, like a tissue culture plate, or are they from like a solid tumor? Each of those are going to require different processing steps to make the cells single-cell suspensions. And in those processing steps, especially for the adherent cells, we need to really be careful to make sure that we don't add back things that will let the cells clump together. And any enzymes we use, we also have to make sure that they don't cleave off our markers of interest. Once you've gone through a process, and I recommend Worthington Biochemical's Tissue Prep Guide, which you can get for free at their website, as a great resource as to processing techniques for different tissues. Then we have to think about the sample prep, going through labeling, etc. If you're going to go to do a sort experiment, it's really important to make sure you filter your samples, especially just before you go into your instrument. The other thing to think about is the concentration. You want to have the cells as concentrated as possible, but not so concentrated that they'll start to adhere and clump together. Adding EDTA to these samples at a low concentration like 1 millimolar is good because it helps chelate divalent cations that also promote adherence to cells. Again, if you're also going to have a lot of cell death, using about 10 units per mil of DNAse will help reduce the amount of DNA that is allowing the cells to clump. So I'd recommend you talk to your core operator and get their feedback as well. But you're really going to start with processing and building that process to make sure you're getting a good single-cell suspension. And don't forget, the microscope is your best friend. So thanks, Joanna, for your question. And remember, if you have more questions, go to getflowtraining.com to learn more about expert cytometry. And if you have a question you want answered here, email it to us at askexcite@expertcytometry.com. Thanks for listening to another Ask Excite. Until next time, be sure to keep up with your best practices in flow cytometry right here with Tim and Team Excite.